Welcome back, students. This is Module 4.1.6, Translation. This is the central dogma, and this is serving as the overview for our current set of modules. Last time we talked about translation, which is the first step of gene expression. You'll recall the way that a gene is expressed is by making a protein. The second step is translation, and this is how we make a protein. And so if we look at translation, it is indeed the second step of gene expression, and we can define it in kind of a brief way as the process by which protein is made from an mRNA transcript. Of course, that transcript results from transcription. Translation occurs in the cytoplasm on ribosomes, and what happens, describing the process, a sequence of nucleotides called codons on an mRNA transcript are translated into a sequence of amino acids by the activity of ribosomal RNA, which is in the ribosomal, and tRNA, transfer RNA. So this is where the three RNAs come together in the process of translation. That's a good thing for you to remember. Before we can really understand translation, we have to study a couple of other things: the genetic code, tRNA, and ribosomes. So first, the genetic code, which is the most complex of the three ideas. The genetic code is how the information in DNA and RNA is read. It turns out, after much study and work, we understand that the genetic code is read 3 bases at a time. 3 bases that are read as one unit in DNA is called a triplet. 3 bases that are read as a unit in RNA is called a codon. And so triplet and codon refer to 3 bases in DNA and RNA, respectively. Now, this triplet or this codon, these 3 bases, they code for one amino acid. The major details of the genetic code were worked out by Francis Crick. And the genetic code is pretty much universal. The broad universality of the genetic code speaks to how closely related all life is. And just to illustrate this point, let's look at a few codons, if you will. We can look at, for example, UGG would be an example of a codon. And we know that this is a codon. It's in RNA because it has U. And you will recall that RNA has the base uracil, and so we know that this is an RNA codon. So what was it? UGG? So if we look it up, U is right here, so the first base, and G, so we know it's in this top part in this row, and then G. So UGG is the amino acid tryptophan. So UGG stands for tryptophan. So there we go. We can practice more. Let's look up AUG. And if we look up AUG, A is the first base, U is the second, and G is the third. It's methionine or start. So methionine is in fact the first amino acid of proteins because it also means to start the process of translation, of making a chain of amino acids. And methionine is the first amino acid. Methionine can appear elsewhere besides at the first position, but it always appears at the first position. So, yeah, start methionine. And so, oops, I'm sorry. We're supposed to look up UGG. Let's look up UGA. So if you look up U and G and A, it says stop. And so it is, in fact, a stop codon. We can look up 4 more: CGA, CGU, CGC, CGG. So CG are the first 2 and look up the last 4. So if we look up C and G, it doesn't matter what the third position is, U, C, A, or G, they're all arginine. And this is showing that there are many more codons than there are amino acids. We have about 64 codons and only 20 amino acids. So we have some redundancy in the code. This kind of redundancy, which makes, for example, the third position immaterial. So it doesn't matter which of the 4 it is. As long as it's CG, if it's U or C or A or G in the third position, then it's always arginine. Sometimes they call that wobble. Okay, it doesn't matter what base is in the third position in this case. And some of the amino acids have wobble in the codons and some don't. Tryptophan, there's only one codon. Methionine, there's only one codon that codes for it. Others have multiples. So triplet we already told you about, codon we already told you about, but a triplet is 3 DNA nucleotides that code for one amino acid. A codon is 3 RNA nucleotides that code for one amino acid. And what is found on that table of the genetic code, it has codons listed, and you can tell because, of course, DNA has T or thymine, but uracil or U replaces T in RNA. And if we look at the genetic code here, we can see that it's U and not T that's present, so we know it's codons that are there. But it's just good to make a note of that. You're not going to have to memorize any codons or amino acids or anything like that. This kind of shows how it works. DNA, remember, remains in the nucleus, kind of like a reference library. If we need information from DNA, we copy it into an mRNA transcript. The mRNA transcript then moves out to the cytoplasm where it associates with the ribosome and is translated, and it's translated into a sequence of amino acids. We can see ACU for threonine, we can see CCU for proline, GAG for glutamate, GAG for glutamate, AAG for lysine. You get the idea of how it works. Okay, so that's the genetic code. Let's talk quickly about tRNA. We already know that mRNA is a type of RNA that copies one gene's worth of DNA into RNA so it can go out and be processed. And so that's the transcript is made out of mRNA. tRNA has a unique function in the process of translation. tRNA has 2 sites that you need to know about. tRNA is kind of shaped like a 3-leaf clover in a way. It has an amino acid binding site and an anticodon site. The anticodon site is actually complementary to various codons. So when we say complementary, remember complementary base pairing. So if it's an A or if it's a G, it will be a C in the anticodon. So you get the idea that it corresponds. And tRNA functions to read the mRNA transcript codon by codon and position the correct amino acid in the chain of a growing protein. There are at least 20 types of tRNAs that correspond to 20 amino acids. Actually, there are more. It's because we have varied codons. There are more than 20 codons. And here is a 3-dimensional model of tRNA, and it's showing the amino acid site down here. It kind of looks like a boot. Near the toe of the boot is where the amino acid attaches. This is shaped in such a way that only one type of amino acid of the 20 will fit there. So it won't take any amino acid, it only fits one. And the fit here at the toe of tRNA is related to the anticodon that we see up at the top. So the anticodon corresponds to a codon. And so whatever amino acid is coded for in the codon and is complementary to the anticodon site, that's the amino acid that will fit down here at the toe of this molecule. And so the codon's an important site. The amino acid site, it has a unique one-of-a-kind fit for only one of the 20 amino acids. And so we have these 20 different tRNAs, each with a unique amino acid site and a corresponding anticodon site. And so this is how the tRNA can read the codons. The anticodon matches up to the codons. And what is the process that allows the anticodon to match up to the codon? It's complementary base pairing. That's what underlies the functioning of DNA. So we're going to leave tRNA now, and we're going to talk about a ribosome. This is the site of protein synthesis. It's the site where the 3 major RNAs come together. Now, a ribosome, this actually shows proteins and ribosomal RNA. The gray is the ribosomal RNA and the yellow is the proteins. And you can see that this is what these little dots or ribosomes are actually made out of. And the ribosome has 2 units, a small subunit and a large subunit. You put it together, and then it has kind of this little tunnel with a little funnel there, these little grooves where molecules such as messenger RNA can fit, and growing chains of proteins and all this kind of stuff. Okay, so we're not going to get into it in great detail here. But let's just do some practicing with the genetic code. The column on the left shows a DNA triplet, then it shows an mRNA codon, then it shows a tRNA anticodon. And so the first example is completely given. We have TAC, and the mRNA codon would be AUG that corresponds to that, and then UAC would correspond to that, and so that's what it would have on the anticodon. And we can work out these others. I'm not going to take time to do it. We'd have to go back and look at the table of the genetic code to figure out the mRNA. Actually, you wouldn't. You can just tell what's complementary. So what's complementary to A is UGG, and then it would be ACC. Right there would be the same. And GAG, we can tell what would be complementary to that. It would be CUC, and then over here again it would be GAG. And in this case, it's exactly the same. The only difference, of course, being if there's a T or a U there. So after a little bit of practice, you could also look at what you'd have to look up on the genetic code is which amino acid corresponds to the various codons. Remember, codon AUG we know is methionine and start, but the other ones I don't remember. I don't have them memorized anymore. Okay, so now after learning a little bit about the genetic code and tRNA and ribosomes, we're ready to undertake the study of translation. So I'm going to give you a list of materials needed for translation, and in the process also give you an analogy by which you can remember this a little bit better. So we're going to need the mRNA transcript that was made during the process of transcription. And the analogy of that, it would be like the blueprint. It has the plans for what we want to build. The ribosome is the site of protein synthesis, and it would be like the factory where we're going to build our product. The tRNAs, at least 20 different kinds, would be the workers. They will read the blueprint and they will bring the raw materials to assemble together. The free amino acids, we're going to link them together into a chain and make a protein. These are the raw materials that the workers would bring. Remember that the tRNAs have an amino acid site, and so they'll carry them over there. Enzymes, of which are many, and we're not going to specifically mention them, they're like tools. Remember, enzymes allow a job to be done, and they themselves are unchanged. Kind of like tightening a nut with a wrench. The wrench itself is not used up. Now, I want you to notice this. You do need to know some of the facts about translation, but I promise that I won't ask you to produce the exact steps of the process on an exam. They're fairly complex. And as a consequence, here I've written them out. We have initiation, elongation, and termination. So I'm not going to read through all this right here, but you can read through it. One thing that you should note and you should learn is translocation, and that's a term that I'll use in explaining what happens here on this picture. So you can see it's really complicated and has lots of stuff, but it's not really that bad. If we just look at the pictures, let's just try to understand the pictures. If we start down here in the bottom left, we can see that this kind of, I don't know, orange or salmon-y sphere is a nucleus, and that double helix, that's a DNA that is going to be transcribed. This little brown structure is the transcript, which leaves to a nuclear pore, goes out, and in the process of initiation binds with a ribosome. And on the ribosome there are 3 sites. They show 2 of them here, an A site, a P site, and then over here there's an E site or an exit site. And so what will happen is that the first binding site for tRNA is right there, and so the first one that comes, notice the first, if we look over here where it says elongation, AUG is the first codon on mRNA, and that's for methionine. So this first amino acid would be methionine, and it would come down here, and this tRNA would sit in this site, and it would bring this amino acid. And then a second binding site right here, it would correspond to GUG, and so this second tRNA would come and it would bring an amino acid, and it would be sitting in the site right next to each other like this. And see this little link here? This is a peptide bond. This is what links amino acids together into proteins, and so that would form under the direction of an enzyme. After that forms, then an enzyme would break the bond between amino acid 1 and tRNA 1. Then the process of translocation would occur, where everything moves down one codon, chunk chunk. It moves down 3 nucleotides, and so now amino acid 2, which was in the second site, moves over to the first site, and leaving the second site available, and so that another tRNA can come in there with the next amino acid, and so forth and so on. That continues on down, chung, 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 chung. And so when that first tRNA is released, the ribosome moves to the next codon position. That's what I'm saying. That's translocation, that movement, and that's how the process of translation occurs. Elongation is just adding amino acid, translocation, adding amino acid, translocation, adding amino acid, and you get this long ribbon of amino acids until you get to a stop codon, and a stop codon will cause the process to stop. You've now reached the end of the mRNA transcript. You've reached the end of your protein, and everything dissociates, and you have your protein, which what might then be further modified after that occurs. The protein is released, and it could then be folded. So this actually shows the whole process. Remember, transcription precedes translation. Transcription occurs. It's the first step of gene expression or protein synthesis. Occurs in the nucleus. Make a copy of 1 gene's worth of DNA into messenger RNA, and then that transcript is processed, take out the non-coding introns, put on a cap and a tail. The transcript then moves out into the cytoplasm, where it associates with a ribosome in the process of translation. And that first step of association we call initiation, and that will start it. And then, of course, after we have elongation and termination, then we'll come out with a final protein that can be used in the cell or transported, and it can be used as is, but most proteins undergo some secondary folding, something like that. So we could rightly expect that to occur. Note over here in this little message here, I've asked you to watch an online animation of protein synthesis. So go to Google and type in online, or type in translation animation or protein synthesis animation, and you get both transcription and translation. I think it's important that you see it. It will give you some insights. There are some, I will warn you, that are very detailed. Again, our coverage here is fairly, we're just skipping over the surface, not going into great detail. So this is kind of interesting. It actually shows the process from top to bottom. A DNA molecule, there's a gene. We have the DNA strand, then we have transcription. Here in red is the RNA, and there's are the codons, and there are the amino acids which correspond to the codons through the process of translation. So what genes code for is for proteins, and that's an important thing to remember. Before we end this module, there's one last topic. It's just 2 slides that we want to talk about, and that's the topic of mutations. I told you when an error is made in replication that is a mutation. And here's a definition of a mutation: a change in the nucleotide sequence of a DNA molecule. And so that occurs, as I said, during replication. Now, mutations are really significant to function. Think of the disease sickle cell anemia. I know we've talked about it several times, but by just substituting one nucleotide for another, it leads to a differently shaped protein, and a protein shape is its destiny. In this case, a misshapen protein, misshapen hemoglobin protein causes your blood cells to be unable to normally carry oxygen, can cause a whole bunch of negative consequences. Now, mutations, of course, then are very significant to function. Mutations are rare, And because we have DNA polymerase, which has a proofreading function, and it corrects most of the errors, only a few mutations make it through. And mutations are also random. They don't occur because of need. If it's getting colder, we don't get more mutations for cold tolerance. That's not how it works. The changes are random. And as a consequence, because they are random, mutations are mostly harmful. If you change something that's working well, say, for example, a watch, take off the back of a watch and see all the nice gears in there, and you jam a pencil in there or something like that, that's a random change, and it's going to most likely be harmful. If you jam it just right, maybe one in 100,000 times or something, it might adjust the watch to make it better, but most of the time it will be harmful. So some few mutations are beneficial, but random changes tend to be harmful. Mutations are also the ultimate source of genetic variation. And so when we talk about genetic variation in cell division, where they come from is here. We talk about reshuffling the deck, but mutation makes new cards. And so the origin of genetic variation is in mutation. And these are mutations, these genetic variations are what is used by biological evolution, and they result in adaptation. On average, there's about 2 to 3 mutations per human genome based on the error rate and the amount of DNA. So that's not very many. But when I look at you guys, I definitely see you as a bunch of mutants, definitely. Okay, just joking. Okay, very quickly, 2 types of mutations. We can talk about a point mutation. These are the changes that are most useful in evolution, where we change one letter of genetic code. We don't delete it. We substitute one base for another. Here is a sentence made out of 3-letter words. They're not all made out of the letters that stand for the nucleotides, but hey, you can only do so much. The sentence says, the cat saw the rat. We might change one letter, change the C to B, and it might be more accurate. The bat saw the rat instead of the cat saw the rat. So it could be beneficial, but if we change the letter randomly, chances are it's going to be nonsensical. The K saw the rat? What's that? We don't know, but that's what's most likely to result from a random change. A second type of mutation is called a frame shift mutation, in which we remove or put in an extra letter or a base. And this actually does something called changing the reading frame. And that's why this is called a frame shift mutation, because you will recall that we read an RNA transcript 3 bases at a time by codons. And if you change one letter, it will shift them all down. It will change all of the codons. And these are devastating mutations. They make everything downstream from it nonsensical. And so if we had the cat saw the rat, if we deleted the C, and now it would be the ats, ought, her, at, oh. And so that, again, everything from there down, all of those would be wrong amino acids, and the protein that would be made would be non-serviceable. And so this is typically a deleterious and lethal mutation because everything is so nonsensical. So with that, we're going to end this module.